use. Nowadays, this is what we do. Because of the base pairing, you don't have to look at both strands of DNA. We just take one strand and we read it. So we look at the data and we try to understand important facts about sequences that we can collect. So one of the main inventors and a visionary in the field is David Hausler from UC Santa Cruz, who had the idea of making a system, what he called a genome browser, which enabled you to actually look at all these messages. And here it is. Here's a genome browser. So this is DNA, and it's not showing the exact letters, but you see here that we're at 219 million, and here 240,000, so on. And there are little annotations all the way along. There are special regions in the DNA which have special properties. So here, these CPG islands have occurred, and they occur in areas methylation, and you can see them just by the frequency of the CG diagram. So it's exactly the same kind of method as we saw when we're code breaking, but at a much larger scale. So the evidence for the CPG islands have enabled us to find what are the promoter regions. It has implications in all of the control of how it works. Here's a nice picture of different Yersinia bacteria, and there's an original one, and then you see the pieces here which have been transposed and moved over. So these genome rearrangements actually can be put under the form of the permutations, just the same type of permutations we were working on, lying a little bit because we do also inversions, so they're signed permutations, but the study of the permutations allows us to see what the dynamics of the inheritance and the genome rearrangements are. I work in an area on the microbiome, as Brian said, and we're interested in certain areas of the bacterial genomes which allow us to identify perfectly certain taxa. So here we have two species of bacteria were particularly important, Streptococcus and Staphylococcus. And when sequencing occurs, one of the things that can occur is pieces of one can go and stick with pieces of another, and they make what are called chimeras. So the methods that we have to use in order to break apart the chimeras and discover them actually are quite inspired by what was used to try and find when there was a break in the wheel order, say, in the Enigma machine. And the toolbox that we use is very much based on using a Markov chain for how the letters follow each other or how the letters can change. And this is this Markov chain between the states A, C, G, and T, which is the same model that Turing used for language. And we also use base factors to try and find important motifs. There's a very good method using the Gibbs sampler that Junlu developed for finding motifs, which use these same factors. And the idea of looking for patterns by hand in the data is certainly something that we use all the time. An effective example, something that I'm publishing recently, it's just about to come out, is work I've done with my group in which we try to find errors, correct the errors, find the chimeras, and then cluster according to particular strains of bacteria, and giving us a much higher resolution than lumping together the bacteria, which is often done nowadays. And so we have a much better idea by modeling the error structure with these Markov models. And our main difficulties. Now, compared to cryptography, we have this problem that we can't say, I see the text, I read the text, I know what the recipe is. When we look at the biology, we say, what should we do now? Well, we have to do an experiment. And so the proof of the pudding is rarely available, and I'd also say that in what we do, the devil's in the details. So I want to thank you, and thanks to the patience of my code breakers, so this is my lab, postdoctoral fellows and students who work with me, and the patience of my collaborators, because we don't always provide by Thursday. I.J. Good is said to have said, I don't want it perfect, I want it Thursday, and we have a hard time providing by Thursday. So thanks very much.
Thank you very much. We have time for a few questions, but there should be some microphones. And
please use the microphone.
So if you have a question, please come to a microphone, because this is being recorded, and the recording can't pick up your voice unless you're in the microphone. I have one question that's only slightly related to your talk, but that's, at the same time that Turing and all were working on the German code, there was also a problem breaking Japanese codes. I think that was done largely in the US, and I was wondering if that also seems very remarkable, and I was wondering if there was communication between those two groups.
They were communicating, and as I said at the early point, one of the main persons, it was William Friedman. So there's this wonderful book called The Man Who Broke Purple, and he broke the Japanese codes, and he was doing it in the US. And so they definitely, I'm very fascinated because of Turing's discovery of the measurement of evidence. I wanted to find out in detail whether Turing had met Shannon and where. And so Turing came to the US 3 times during the war, and he'd come before the war. He'd come to Princeton, so he'd come to New Jersey, and he met with Shannon several times. It's said in the book, very good book on Turing by Hodges, that they had several meals together, and they only talked about voice transmission and voice recognition. That is, they didn't talk. I'm very interested because, in fact, Shannon went on. He did log base 2 for information theory, and it seems that Turing had done the log base 10. That it was doing the bands, but the ideas are very, very close. So they did communicate, and in the US they were making bomb machines also to try and help because they had more resources. The British were strapped for steel and all kinds of machinery, so they did communicate. They were working together. Yeah.
Did the Germans ever work on breaking the British or American?
The Germans did work on breaking the British codes, and it's very well reported on. In Friedrich Bauer was a German. He didn't work during the war, but he knew very, very well what was going on, and they did work on them, but they didn't have the same success as the Allies had with the Enigma. And I think the Allies were much more paranoid in some sense. The Germans were quite arrogant. They were absolutely sure that the Allies couldn't read their coded messages. They were sure that they had this mechanical one-time pad and that it wasn't breakable. And that confidence is in some sense what lost them, because they kept on using the same machines. They changed the protocol a little bit, but they had they kept going. And I think that the Allies had many different systems. I don't know if between Silk and Cyanide is a wonderful book about one-time pads, and they had silk handkerchiefs with one-time pads written on them. So they had poem codes. There were lots of different codes. Yes, I will repeat your question.
The other question. You showed us a very fascinating transition from human code to code used by
Yeah.
question. Is biological nature good at coding? I mean, are codes straightforward or is
So the question is, are biological codes straightforward? I would say no, given the length of the human genome. If you had to put that amount of information, encoded, for instance, in a book, Craig Venter's genome would be volumes and volumes and volumes, which would take up this whole stage. So in some sense, it's not efficient, but we don't know all the stories. That is, for a long time, there was the impression that within the genome, for instance, there was the coding region, and then there was what people called the junk DNA. And all of a sudden, we decided it's not junk. There's a whole lot of information in there that we just weren't aware of because we don't know. So we are very far from understanding the coding. I mean, the brain, we don't understand that either. So but I do think that listening to all the evidence and taking everything into account,
殺された。 プ落としました。ストップ落とした。すみません。どうも。すみません。ありがとうございます。上級コースをスイスイやられるように10年ぐらいかかるから。5年ぐらいか。 これ4月5月までできそうだな。